So it's a real pleasure for me to introduce Sumit Arora, who will be giving the presentation today. Sumit did his master's and PhD at NIPA in India, and as some of you may know, previously worked at Simcyp from 2018 to 2021, where he was involved in the dermal and oral project teams there. Unfortunately, Sumit left in 2021, and now he is at Janssen Pharmaceuticals, working in the biopharmaceutics team there. So, Sumit, over to you.
Thank you, James, for the kind introduction, and thank you for the organizers for inviting me to give this talk. This work has already been published. And the link is there. So if you want to go and have a more deep dive into this work, I would be happy to take any questions, and please feel free to contact me afterwards as well. So really, as James said, this was the work which I did along with the whole dermal team at Simcyp. And what we were trying to achieve there is how can we use in vitro data more effectively to inform dermal PBPK models, and can we develop IVIVE tools which can be used for prospective predictions of how the drug is going to be in different layers of skin. So that's really the start of the work which we tried to did during my time at Certara, and James and the others are continuing this work. So in this presentation, I would like to give you a bit of a background about IVIVE and what does it mean for a dermal drug delivery. We're going to talk a bit about the formulations, because topical formulations, though it appears simple, are quite challenging to model. And that's because these formulations undergo metamorphosis when you apply these drug products on the skin surface. We're going to talk a bit about the PBPK model structures and what kind of input parameters you need for these models to parameterize these models. And finally, I'll share a case example of Metronidazole. And I'm happy to take any questions you have during the presentations or after the presentation. So the motto for this work is really, the philosophy remains the same, whether it is modeling drug-drug interactions or modeling absorption from oral administrations. It's really understanding in vitro to predict in vivo. IVIVE is not a new field. It's been quite established in the field of drug-drug interactions, where people are trying to use microsomes or hepatic enzymes and other systems to understand how can we generate in vitro data and then use appropriate scaling factors to then able to predict what's going to happen in vivo. So it's the same set of philosophy which we apply to the dermal drug delivery. And to go in a bit more detail, what does we mean by dermal IVIV is that? So I want to focus you on this one. We routinely do these kinds of experiments when we are trying to develop topical products. So in vitro release testing, IVRT, or in vitro permeation studies, or IVPT, that's quite routine. So companies generate a lot of these data to try to understand how their formulations, and if they change something in their formulation, how that's going to affect the permeation of the active compound. So this is quite routinely done. And it's important that we start looking at these experiments from the modeling perspective as well, because they can give us quite good insights how to predict the drug behavior in vivo. These studies, when combined with more understanding of your drug product, so Q1, Q2, Q3 means your qualitative, quantitative, and your microstructure properties of your topical drug products. For instance, if it is a gel composition, your solubility of the compound in different phases. If it's an emulsion, then you would like to know what's the drug is in the continuous phase versus the dispersed phase. We're going to talk about metamorphosis in a minute, and then you will understand how this drying rate or weight loss or the evaporation of the volatile components from the drug product is important to understand, how the rheology of the product is changing, or if your drug is precipitating on the skin surface. So we really need to understand what's going on with our formulations when we apply it on the surface of the skin. The idea here is that you combine this detailed characterization of your formulation with your IVRT or IVPT studies. And in this model, this is a model of the skin. So for IVPT, you can actually replace this model with a receptor. So the top layers remains the same, but the bottom can be, because these are the blood flows, you don't have blood flows in IVPT. So you can replace that with a receptor And you can try to model your IVPT experiments, because it's more like a confirm and a learn cycle, because some of the parameters which you put in your model are always uncertain. So you would try to understand how to gain certainty in those parameters, and modeling in vitro experiments can provide certainty in some key input parameters. And once you know that your model can pick up your IVRT or IVPT nicely, then you can replace the bottom part or the back end with the more physiological part, and therefore try to understand how your drug is diffusing through the skin surface and try predicting local concentrations. So that's the whole philosophy of thermal IVIVE. And I'm gonna show you an example how did we did that for metronidazole drug product. Just to quickly classify, all the dermal products are either solutions or lotions or semi-solid formulations or suspensions. And all of these can be categorized basically into three categories as solutions, emulsions, or suspensions. So gels and creams are usually when you have quite a lot of water present in your formulations greater than 50%, or ointments or paste when the water concentration is less than 15%. But overall, all the different formulations which are available out there to apply on the skin surface can be broadly classified as solution, emulsions, and suspensions. So I briefly mentioned before about metamorphosis of topical formulations, and I want to spend a bit more time. What do you mean by that? So you can see it, the product, the formulation which comes out of the container as you apply it on the surface of the skin is something looks like this. But immediately during the applications and when you are starting to rub this product on the surface of the skin, the morphology of the product changes completely. So a lot is happening during these phases, and we need to understand and characterize all these processes. So you can see the initial composition of a formulation can look something like this, that you have a lot of water initially from these drug products when you start applying it, but however, at the end of your application, your product composition could be some completely different. So we need to understand this, what's happening during this process, and try to model this. This is an example of betamethasone dipropionate and calcipotriene product from Leo Pharma. And you can see that initially the formulation is coming out from a pressurized container, and there is a quick evaporation of your propellants. So your concentration of your actives is somewhere in this range when it's in the container, but as soon as the actives are evaporating, the concentration goes up because you are making a more concentrated solution. So your drug products can end up in a supersaturated state, and this could really enhance the concentration gradient of your drug, and therefore your drug may permeate more through the skin surfaces. So understanding of these processes are quite important to put into the models so that we can predict their in vivo local exposure. This is just a graphic showing different kinds of processes which may go when you are applying a solution or a monophasic gel or an emulsion or a suspension. So you can see when you apply even a solution, a lot is going on. So in the model, in the Simcyp MechDermA model, what we try to model is how much is the drug is diffusing from the continuous phase of the formulation. So your drug diffuses from the continuous phase to the stratum corneum, and you have to take into account how the vehicle is evaporating, if there is any changes in drug solubility and vehicle viscosity as your product is evaporating on the surface of the skin. So all these processes needs to be captured in order to predict the exposure in the skin. For emulsions or creams, we need to understand how the drug distributes between the oil and the continuous phase. It's still the continuous phase where we model how the drug is getting absorbed, unless this has changed now in the recent version. But that's the assumption at that time when I was working with Simcyp, that it's really the drug coming from the continuous phase, which goes into the skin surface. We can also model solid particles. So if your drug is present as a suspension or as a solid particle, the dissolution of these particles can be modeled, which is then continuously feeding the continuous phase for the drug to get absorbed. So all these processes are needed in order to capture in vivo exposure. This is a cartoon which I took from a presentations from MCERC. This is the multi-layer, multi-phase MechDermA model. And you can see it from there that You have a compartment to model formulation, so you can model all the processes which are going on once we apply the drug product on the surface of the skin. Stratum corneum is being modeled as a brick and a mortar. So your corneocytes are brick, and the lipid matrix around these corneocytes are modeled as a mortar. The model considers an effective diffusion coefficient across the skin surface. It also takes into account the tortuosity of the skin surface. Afterwards, the stratum corneum, the viable epidermis, dermis, subcutaneous, and deep tissues can be modeled. The drug can be absorbed into the systemic circulations via dermis, subcutaneous, and deep tissue. A clearance can also be defined if there is a metabolic clearance happening in different layers of the skin, that could also be defined in the model. And these models are then parameterized based on different populations. So the model has been parameterized for pediatric. There is an ontogeny functions built there. North European, Caucasian, elderly, ethnic, and a disease population as well. And in additions, you can imagine that the skin on your hand could be completely different from the skin on your forehead. So there are different application sites within the MPML MechDermA model that one can use to approximate the physiology of that particular site. So that's in a nutshell the MPML MechDermA model. And what kind of input parameters you then need. So it's a busy slide, and you need quite a lot of parameters for these models to work. However, a lot of system parameters are provided by the software itself. And for an in vitro simulations, you can either simulate a static or a flow-through cell. You can select what's the skin anatomical region you want to simulate, either back or forehead or those kinds of an anatomical region. You can define the thickness of the skin sample, what's the area of the diffusion cells, volume of receptor fluid. For in vivo simulations, of course, you replace all these parameters with the underlying physiology of viable epidermis, dermis, and further tissues. You need to define the trial design as an input parameter. So number of subjects, demographic, what's the age, range, and gender, what is the dose and the volume of the formulation applied, and for what duration do you want to run the simulations. So for your duration of action of your drug product on the skin surface. Basic physical parameters are needed for the drug, such as molecular weight, log P, pKa, and a lot of partitions and diffusion coefficients through different layers of skins are actually predicted by QSARs. QSARs really help you to provide you a starting point, but you need to be careful in terms of what are the data sets which are used to build these QSARs, because each QSARs can have some of its limitations. But that's where we need IVPT kind of data to understand and validate these predictions coming from QSARs. And we're going to show you the workflow. How can you use in vitro permeation data to gain confidence in these partitions and diffusion coefficients. And finally, you need information about your formulations. So what's the composition? What is the drying rate? If there are any changes happening in the rheology, if the drug is getting precipitated. So all those kind of understanding is needed to put everything together. So I'm now going to show you how we used this workflow of dermal IVIVE to predict in vivo skin permeation of metronidazole from two commercial products, MetroGel and MetroCream. This is the workflow which we adopted, and I'm going to take you through this one in a minute. But this is the workflow which we adopted for modeling metronidazole exposure in the skin. So we started with collecting of physicochemical parameters of metronidazoles like molecular weight, log P, pKas. We use QSARs as I indicated initially, and there is a lot of research ongoing that what QSARs are the most appropriate to use as a starting point. So that's really a big piece of work which we were doing at that time, and hopefully still ongoing. There is also skin anatomical and physiological parameters. Depending upon where the tissue sample was taken, you have to define the skin physiology. And then initially we started with IVPT data of metronidazole from aqueous solution. So we started with something really simple. So if you are thinking about creams and gels, there are much more complex formulations. And if you start immediately with these kinds of formulations, you may end up with certain parameter identifiability issues. So if it is possible that you have data just from simple solutions, I know it can be challenging for lipophilic compounds. But if it is possible that if you have certain data from simple solutions, start there, because that's really removes a lot of uncertainties which can creep into your model from if you just start from complex formulations. So we started with something really simple. I say simple, it's not that simple, and I'll show you in a minute. So we start with a solution, an infinite and a finite dose. So we start with even an infinite dose where we know that the drug concentration stays on the surface of the skin and there is no evaporation happening, nothing, those kinds of things, so that we can start building this model bottom up. So we started with solutions, we parameterize our PBPK model, and then we evaluated. Our evaluation criteria at that time was that if the predictions are within 2 folds, yeah, it seems to work. But if it is outside 2 folds, then something is not right in the model. So if your model predicts well, yes, you can. Yeah, if your model explains solution data, you can start parameterizing your model with the properties of your formulation. But if the model does not predict, then you have to go and find out which of the parameters you need to identify which of the parameters to optimize in order to predict your solution data. And it's also an interesting one that it doesn't come easy that which parameter to optimize, because multiple parameters can give you still the same result. So it's quite a lot of iterations which has to be done in order to identify the right parameter to optimize, and of course scientifically justified. So once you know your model is predicting your solutions, you start integrating your Q1, Q2, Q3 properties of your formulations. You then evaluate your model. If it doesn't predict, you go back and see, okay, what changed between a solution and a formulation, and then you start identifying other parameters to optimize. We are still using in vitro data here. So we are not trying to use in vivo data to optimize predictions. We are still trying to see, can we find a way to use our in vitro data efficiently to predict in vivo. So once you think, yeah, the model can capture your formulations, you then can either challenge your model with a different formulations. We did in this case, once we know that our model was able to predict MetroGel, we challenged the model with another formulation, Metro Cream. And once you know your model is predictive of your in vitro experiments, you then replace your, you then put in your in vivo physiology and try to see if you can predict local concentrations in vivo. And of course, at this stage, we always do extensive parameter sensitivity analysis of all the CQAs or quality attributes or CBAs, critical to bioavailability attributes, which is impacting the performance of the drug product. So we started with MetroGel. We initially started with infinite and finite dose solution from IVPT data generated from solution. We have quite extensive characterization of MetroGel in terms of pH, viscosity, evaporation profile, drug solubility. We not only had the data from one lab, we actually had data from 3 labs. Same data generated between different labs, characterization of QT generated between different labs, because we wanted to understand how does the data generated in one lab translates to another. We have IVPT data for 3 different doses. So we actually tested 3, 10. So this was tested by Murthy group at 3 dose levels. We also had the IVPT data from University of Queensland, from Professor Mike Roberts group, and then we searched for the literature. I'll come to one of the shortcomings of these kinds of modeling, because there is really a scarcity of data where what you do actually in vitro, you match 1-to-1 in vivo. So those kinds of data sets are missing, and there is a need to build those kinds of data sets that if you apply a certain amount of your formulations on an in vitro setup in a certain area under certain conditions, you need to replicate that in vivo so that you can make one-to-one translations, because changes in these conditions makes IVIVE a bit tricky. And more data sets are needed where you need to match your in vitro conditions to your in vivo, just to understand this a bit better. So we started with modeling infinite dose experiments from solutions, and you can see this is the observed data. And just with using QSARs, which are predicted, you can see the blue is the mean, and we were completely under predicting. So something was not right in the model. There are two parameters which you can optimize in order to fit. Either you can optimize partition coefficient, so how your drug is partitioning from your formulation to the lipids, or you can optimize your diffusion coefficient. So we tried both. So if you optimize diffusion coefficient, what we end up that in order to capture what we are predicting here, you have to increase your diffusion coefficient to a value where it's basically diffusion in water. So you have to raise your diffusion coefficient to a very high value, which is not realistic because your compound will be diffusing through lipids, and just optimizing it to a value of diffusion coefficient which is actually diffusion in water will not make sense. So we looked at the data. We looked at the QSAR, and metronidazole is a hydrophilic drug. And if you look at the QSARs, they are actually, while building these QSARs, there is quite a lot number of compounds which are lipophilic, and there are really small data set of the hydrophilic compounds. So the data set's a bit biased, and there is, of course, it's challenging to quantify for the hydrophilic compounds. So there was quite little data, and we know that KP could be mispredicted for hydrophilic compounds. So this was the reason why we choose KP lipid to water, and we optimize this parameter. The idea of the whole exercise is if you use a certain data set to optimize a certain parameters, you try to fix that parameter. And you only change it if there is a strong rationale to change it, but you don't change your parameters from data set to data set. Otherwise, there is no consistency in your modeling work. So we optimized KSC lipid to water to capture the permeation from the infinite dosing conditions. So this was fitted. We then try to explain this from an finite conditions, again from the aqueous, and this was able to capture. So once you use the infinite dose data to put your KP-SC lipid to water, we were able to explain finite dose experiment from the aqueous solution. This is the concentration which Professor Murthy measured in their lab from the aqueous solution. So we fitted a first order evaporation kinetics. We put that into the model, and the model was able to capture the permeation profile. So that gave us a bit of a confidence that the fitted parameter of KP was verified with an additional data set of finite dose condition. So we were happy that, okay, now our infinite and finite dose aqueous solutions are working. We then went on and parameterized the model with MetroGel with all the Q3 properties. And then we realized, okay, this was our observed data and we were a lot over predicting. So clearly something is still not working in the model. So it's not that easy to then translate what you are observing from solution to that of a cream. And we went back and have a look like, okay, what's going on here now? And when we looked closely at the experimental conditions, what we found out is that in these IVPT experiments, these experiments are actually done with a large volume of formulation or for a large volume of an aqueous solution. So around 2 ml formulation or 2 ml of aqueous solution was used in an infinite dosing conditions, and in a finite was around 300 microliter. However, in the IVPT experiments from gel, we use around 10 microliters. So there is a huge difference in the amount of or the volume of the water which you are putting on the surface of the skin in these experiments. And we went back into the literature and look, okay, can this create a difference? And we found the publications from from Yusuf et al. It's from University of Queensland, where they looked at how skin hydration changes. So if you put a large amount of water on the surface of the skin, you are changing the hydration of the upper layers of stratum corneum, and this could have an effect on the diffusion coefficient of your drug. So they found out that the hydration has an influence on the diffusion coefficient of the drug. It does not influence its partition coefficients. And clearly in these experiments where we were using infinite and finite, there was quite a large amount of water, and therefore our diffusion coefficient was able to predict. But however, for formulation, that was not working. So based on this skin hydration, and we know skin hydration affects diffusion coefficients, we try to optimize the model again for the diffusion coefficient, which is specific for the MetroGel with this 10 microliter. And once we optimize it with a 10 milligram per centimeter square, we were able to capture it. So if you optimize it, of course, it will able to fit. However, the interesting part comes, again, you always need to have independent data sets to challenge your model. So the data sets which you use to optimize your models, there should always be an independent data sets where we need to challenge these models. So this was able to capture this one by fitting. However, we then use because in here we are using 3 different doses. So you are putting different amounts of your drug on the surface of the skin, different volumes of formulations. And it was characterized in vitro that the 3 milligram, that the evaporation potential of 3 milligram and 30 milligram are different than 10 milligram. Because you are putting a bit more drug, it takes a bit longer to evaporate. So we used the Q3 data of those evaporations profiles and fitted and provided those inputs to the models. And if you fit the model to 10 milligram, we were able to explain 3 milligram and 30 milligram. And you can see the exposures are a bit different. You have much lower exposure here, is a bit higher exposure here. So in this way, we were able to confirm that, okay, if we even have to optimize diffusion coefficients, our model was able to pick up 3 milligram per centimeter square exposure and 30 milligram per centimeter square. So the model was able to capture metamorphosis of formulation.
Hello, Samit. Hello.
Yeah.
Sorry, this is Ali. So you tried to fit the diffusion coefficient previously, and then you said the diffusion coefficient was very large, to the extent of the diffusion of the compound in the water. What did you find here? And basically, you did it like a stepwise. So you first fitted the KP, and then you fitted the diffusion coefficient. Is that correct?
That's correct. Yes.
So could you do simultaneously 2 fitting at the beginning, and was it different?
Yes, because that was solution. So if you have to, you can find a bit of a intermediate parameters, that's also possible, but we didn't try to do that simultaneously. But that's something could be tried.
And how much did you get for here? Was it way below than the diffusion?
Yeah. Because you have to, because right now with this diffusion coefficient, which was in the lipids, you have to lower your diffusion coefficient in order to capture, because earlier we were under predicting. So we have to increase the value of diffusion coefficients. In this case, we were actually over predicting. So you have to lower the value of diffusion coefficient.
So it's because you previously you didn't fitted the KP.
Yes.
Now you fitted the KP, and now you're over predicting.
Yeah. So
you had to fit the diffusion coefficient to lower it down again.
Yes, for this case. But we found out that this could be one of the cases from skin hydrations, because there is actually a difference in the volumes, how you apply and how much water you apply on the skin surface. So this needs to be taken into account.
Okay.
Yeah.
Thank you.
But here the idea is that if you can explain 10 milligram per centimeter square, you should be able to explain the other doses as well. Because the metamorphosis is happening differently from these doses. The evaporation rates are different from these doses, and the model should be able to capture this. So we were able to then verify the other 2 doses with 3 and 30. We also have, you can see these are the 2 different evaporation profiles, one from the Murthy lab and the other from the Roberts lab. And we then used the data which was generated in another lab. So the data which I showed you previously was from Murthy, And then when we parameterized the evaporation profile using the Roberts, we were able to capture the exposures which was observed in Roberts' lab. So we were able to then predict the exposure from a different laboratory with the evaporation profile determined in that particular laboratory. So that gives a bit more confidence. Okay, the model can capture data coming from different labs depending upon how the experiments are done in those different labs. It also raises an awareness that you really need to be conscious about what kind of data sets are you using to validate your model, and you characterize and do all your characterizations in a similar kind of an environment, and just don't take data sets from different sources and combine it and say, yeah, my model is working. So that's need to be considered as well. These are the results of the parameter sensitivity analysis. Of course, for gel, evaporation rate was a very sensitive parameter. So the estimates which we got from the in vitro data, that was able to predict. But if you can, and this also gives you a bit of an understanding how you can use these tools to further optimize your formulation. So if I can add any agent which slows down my evaporation, or which fasters my evaporation, then you build really super saturated solution. And that's what Leo Pharma did in their product, where I showed you this betamethasone and calcipotriene profiles, that they made their solution in a propellant which evaporates faster, and therefore there is a super saturated drug product on your surface. So you can actually promote skin permeation. So you can use these models to understand, okay, what kind of evaporation rate should I aim at to get enhanced permeation. The model was sensitive to this. The model was also sensitive to precipitation rate constant. So if your drug is precipitating, of course, you are reducing the soluble concentrations which can diffuse. So if you can add certain inhibitors, which can again lead to supersaturations, more concentration of the drug can go into the skin surface. So model was sensitive to PRC as well. We didn't find much sensitivity to vehicle viscosity, and there was also some limited sensitivity to the drug solubility in the continuous phase. These are the observed metronidazole values, which was reported in literature. And now when we know that, okay, our model was able to predict in vitro, we then went on and did some in vivo predictions. There are certain assumptions, and I previously mentioned you need to have very consistent data sets, that whatever experiments you are doing in vitro, if you can have similar data sets generated in vivo, that is the ideal data set to use for these kinds of modeling. However, we have to make certain assumptions for in vivo. So we got data from the Rosex, and we assume that this is similar to MetroGel because both of these are 0.75% weight by weight gels, and their compositions are quite similar. And you can see the data which is reported by Dykes and Azubero. The model was able to capture this quite nicely. Of course, there is huge variability in the clinical data sets. That's something is there, but at least you are in the similar ballpark range of these predictions. And this was just done bottom up. So we optimized our model with certain parameters from using in vitro data, but when we were trying to predict in vivo, and this was within 2 folds what we observed. As a further verification of the model, we challenged the model within another formulation. So you can see we started with something very simple like solution. We then went on to a bit more complex formulation, which is a gel. And then we finally challenged the model with a more complex formulation where you have an oil and a continuous phase. And we challenged the model with this Metro cream. So this was also 0.75% weight by weight cream. This should be cream. We also had a whole battery of Q3 characterizations, pH of the formulation, viscosity, how does the evaporation profile of this product look like, what is the solubility in continuous versus dispersed phase, and we have an IVPT data from 3 different laboratories at 10 milligram per centimeter square. And when we parameterize, so we didn't change any of the parameters in this exercise. So whatever we optimized for K diffusion and partitions that were kept the same. Only the formulation properties were inputted as a model parameter. And we use the evaporation profile of the creams, and you can see these are our predictions. So the squares are from the Roberts lab, the circles are from the Murthy, and the triangles are from the Zhang lab. And we were able to predict this for the cream for using the Murthy QE and using the Roberts Q3 properties. Actually, the evaporation profiles in both the labs are quite similar, so the predictions were actually quite similar. So in this case, we were able to predict performance from a cream formulations by using the Q3 characterization from the cream formulation. So in this way, you can probably appreciate that this IVIVE can be very helpful in dermal drug development. This is, of course, initial modeling and trying to develop a workflow that how should we approach modeling for dermal drug products, what kind of data sets we need to gain confidence in our modeling approach. So this is really limited and is a first example in this space that how can you approach this. But of course, there is definitely a need to further verify this approach and probably challenge this. There can be other ways as well, but it's just to promote sensitivity in the community that there is a way that you can use a lot of your in vitro data, which you are anyways generating to develop the formulations, and how can you use that data to then build models which can be in vivo predictive. These models can also be used to understand differences between a reference and a test product. So they can be used to generate bioequivalence for a reference and a test products, which is actually quite interesting because it's not so easy to develop generic topical products for skin. And of course, we demonstrated in this case IVIVE for MetroGel. And I was explaining that you definitely need consistent data sets between how you apply the dose in vitro to that you apply in vivo to further gain confidence that you can reliably predict in vivo exposure from your in vitro verified models. So I think that's all from me, and I'm happy to take any questions if you have.
Sorry, again, it's Ali. Can you go back to the slide you were showing the precipitation stuff?
Yeah.
Yeah. So what kind of experiments are out there for, I mean, just to quantify these these precipitations rate constant? Did you just get the values PRC fitted based on your concentration in the your in vitro experiment, IVPT?
Yeah, in this case, metronidazole is a highly precipitating drug. What you can do is maybe simpler experiments to understand this. In this case, what we found out is that metronidazole is very soluble. So as soon as it goes slightly above its supersaturation, it immediately precipitates. So what we assumed in this model is that as soon as your equilibrium solubility overshoots, you use a large value of precipitation constant to keep your drug in a solution state. So whatever, as soon as it goes, it just precipitates, and that's what worked in this model.
So I assume because the reason for precipitation here is the evaporation of the formulation. So I assume between different formulation, these PRC values should be fitted separately. You won't be able to use for the same value for different formulation, would you?
Yes. Depending upon how the formulation. In this case, since we know it's a hydrophilic drug, the same PRC worked for gels and for creams because the model assumes that it is the continuous phase where the drug is getting absorbed. So in the continuous phase of the cream is also water.
Right.
And therefore the driving concentrations in the same phases were assumed constant. But you are right. If you have certain ingredients, then you probably have to, and I'm just thinking on top of my head now, you have to make certain solutions with different viscosities and try to understand, okay, what is the solubilized concentration there, and can you estimate a precipitation constant, because you do have to relate it with your supersaturation index or in your formulation as your vehicle is evaporating.
Thank you.
I don't know if people on the call have more experience now for other drug products.
I don't think there's any more insights than that. In for any other drug products from that we've come across since this case study Sumit.
Yeah, but it's a valid point, Ali. Yeah, depending upon how you are making these formulations, your precipitations can be. So that's why you need all those in vitro experiments to parameterize the model.
So that's why I asked if you have any other source of experiments, like you can quantify these precipitated drugs on the surface of the skin using a kind of I don't know what source of imaging experiment or something.
Yeah. The one which comes to my head is using different viscosities, because you also have to relate it with the viscosity as well. And that could also reduce your diffusion coefficients and maybe your precipitation rate, because the vehicle is evaporating, your drug product on the surface of the scale is getting more concentrated, your viscosity is increasing. So you probably have to replicate some of these conditions in vitro to try to understand how to parameterize this.
Yeah.
In this case, from the FISCAM, we know that it precipitates really fast. So we assumed a very high precipitation rate constant, and that worked across both gels and creams.
Hi, Sumit. So, Divyan here. So basically, if we see the viscosity impact or any other Q3 characterization impact during method validation only, the CROs or during the experimentation for the validation purpose, it will be required to have different viscosity, right? So it will be basically always going out to be a part.
Yeah. You have to, but at least from your initial sensitivity analysis, you can at least pinpoint what kind of experiments you need to do to gain further confirmations, or you can make some hypothesis like, okay, this can be an important parameter, then you design certain kill experiments to either support or kill that hypothesis. So that's really, I find it useful with these kinds of parameter sensitivity analysis, that at least you can pinpoint, okay, for this case, yeah, this is not important, it's not giving any difference, but while these can be important parameters, and then you go and design certain experiments.
Yeah, thanks. So I have one question. It is more practical in terms of because I had seen certain studies. So basically, for the when it is a disease condition, we are asking the label claims that apply a P shape or something, a small amount to the affected area. But when we are going ahead for a PK study, at that time, most of the time, due to the lower bioavailability, we would like to have it on a larger surface area, like 1000 centimeters square or 500 centimeters square, with 5 gram or 10 gram as a quantity. So how or what practical difference it could make? Any idea?
Yeah, you have to match, like you can of course scale it down. You have to match your milligram per centimeter square applied to your in vitro. So in that case, that can be matched. You don't have to always go. Of course, from exposure point of view, you need that in an in vivo, but probably for in vitro, you can get away with milligram per centimeter square.
No, I'm asking more for the in vivo because the LLOQ is really critical, and at that time, like for many drugs are having a 1% or 2% viability. At that time, like it is going out to be a must to have a larger application or of a quantity or and a larger area, like whatever the procedure we want to say, rub it for 10 minutes or 5 minutes and then standardize it. But at least the quantity is going out to be very huge.
But your concern is that if you are using a huge quantity versus a small quantity, does it makes a difference?
Yeah, because when it is really affected, if we see the real case scenarios, it is going out to be applied to a very small area of the affected disease area only. While for the PK purpose, we are doing it on a quite large quantity with a large surface area, like back or uh,
Yeah.
other areas. So, do you think it could have any impact, or for just meeting the LLOQ, we have to have a higher quantity?
Oh, you probably have to go back to your purpose of study. Are you developing a novel drug product to understand how it's going to, or to characterize its PK, or if it is a novel drug product, or are you trying to develop a test or a or a generic drug product? Because then the concentrations, I believe, would be different. Because right now, what you're concerned is if you apply to a large surface, which is a small surface, uh, the good point of the model is that you can always quantify stuff. Whether you can quantify, because you are going for a large surface because of your experimental um, uh, because of your experimental limitation, that your LLOQ or your quantification limit is so low. So you probably, when you are trying to simulate that study in a simulator, I know simulator was trying to be expanded at that time for MUST studies, and you can expand that surface. So that is as far as if you are applying milligram per centimeter square and you are kind of normalizing, I don't expect that you would see a difference.
Okay, thanks. So, one more last question. Like, basically, if we see the drugs which are, what I can say is like, uh, I am not able to express. No worries. Thank you.
No worries. Please send me a question in case you want to discuss this a bit more.
Yeah, sorry, I got it. So, it is a weird question, I think so at least. So when we go ahead for the IVRT, IVPT, and then IVIVE, normally the most challenging part is the IVPT. And many a times due to the donor to donor variability, there are a lot of challenges with respect to IVPT. Is there any possibility to explore the modeling for this purpose to justify that the variability which is there, it is not it is due to the drug substance itself, and not, it doesn't have any impact of the in vitro method or anything else, and the data can be used, I mean, we can use the large variability of IVPT still?
Yeah, that's always a concern, and I think we were looking at this during my time at Simcyp, and probably James could also comment on this. Well, we were looking for certain kind of a characterization, that if you can do certain kind of characterizations, maybe limited characterizations of your skin surface, which went into the IVPT studies prior to doing those, like for instance, in terms of tissue depths and in terms of analyzing, if you can do that kind of information, then you can make that physiology in the model. So I don't know how easy or difficult it would be for the CRO to a bit more characterize the skin surface, because we also identified that as a gap, that how are you making sure that the physiology which you are generating with your simulator is actually representative of the physiology which went into the IVPT experiments. So certain characterization of the skin, if that's possible, that could also help you in refining that physiology to match. Then you are also comparing apples to apples and not something else.
Yeah, thank you, that's it.
But I don't know, James, if something were done or how this been approached now.
No, I mean, we still don't have access to that kind of data, unfortunately. But one of the things that's interesting if you kind of if you look at inter and intra donor variability, so if you've got, because a lot of the time you'll have, say, 3 or 4 replicates from the same donor, which is from the same piece of skin, like an adjacent piece of skin, but you still have huge variability there between just those replicates. And so, like, the analysis of that says that actually most of the variability in the IVPT studies isn't physiological. because you've still got that huge variability, which implies that a lot of it is experimental. And so then that's really difficult for us to pick up in a kind of mechanistic, physiologically based model that has to be tackled from the top down, really.
Yeah, but I think you probably then have to stick with a similar lab that they are sourcing their skin, and the experimental conditions at least are optimized for that, so that at least you make consistency between all your data sets. That even if you have to fit certain parameters to capture it, but your formulation behaviors are still adequately explained. Yeah.
That's all the questions. The only question in the chat was from Divyan, and he's asked that already. Does anybody else have any further questions for Sumit? Okay, well, thank you very much, Sumit. That was really, really great. And thank you everyone for joining.
Thank you, everyone. Bye-bye.
Yeah, take care. Bye-bye.
Thanks, Sumit.